I was trained as a clinical neuroscientist, and the way for me to sort of understand the brain in pathology was to use images of the brain. And for me, my sort of interest and research focuses on obtaining and analyzing data coming from these big machines that you often have seen in USC facilities. So here on the left, you have this big scanner, this what we call a magnetic resonance imaging or MRI scanner. And here on the right, we have the positron emission tomography or PET scanner. These are pretty big pieces of equipment. Sometimes they make a lot of noise. You probably you've seen them before, and they're at USC. The scanners themselves, for me, they're sort of amazing pieces of engineering. They're really complicated machines, and their purpose is to take images of our brain, and together they provide sort of information about the different parts of the brain, and using that information, we can sort of, they can help us understand neuropathology in the brain, and specifically here, how it relates to Alzheimer's and dementia. So starting with MRI, the magnetic resonance imaging, I find it quite fascinating. I think it's almost sort of a very nice physics experiment. And one way to understand what is happening when you go into the MRI scanner is to think of our brains as basically big pools of water. So our brains contain tons of water, and the water contains protons that one can think of them as little magnets. And when you go into the MRI scanner, this scanner is basically a humongous magnet. If you compare it to the magnetic field of the Earth, is about maybe actually even more than 30,000 times stronger than the magnetic field of the Earth. So it's a pretty big magnet. And what happens is that when we put our brains into the scanner, the sort of little magnets in our heads align with the magnetic field, and with then the sort of when we apply a specific excitation pulse, what we do is we basically make these little magnets rotate, and then the sort of coil that you have around your head is like a little receiving coil that basically collects the signal from these magnets. And this is how we're actually able to collect data from our brains once we go inside the scanner. So what kind of images can we get from the MRI scanner? So depending on how we collect the signal, and that's actually quite important because the different tissues in our brain have different magnetic properties, then we can collect different types of images that in essence emphasize, i.e. they give high signal, or de-emphasize, i.e. give sort of less signal in different parts of the brain. So for example, one particular image that we're getting, which we call the T1 weighted image, is an image that basically outlines the gray matter, which is this little ribbon right here as gray, so we can see what the gray matter is in the brain, and the white matter, this sort of big white area here in the middle as white, as well as the cerebrospinal fluid being outlined as dark, right? So here we can see the gray matter, the white matter, and the CSF basically using different colors in that image. And I want to emphasize that although I'm presenting a slice of that image, the images that we're getting are actually three-dimensional. So you can imagine that we can rotate that image and see every aspect of the brain. And again, if we apply a different kind of sequence in the scanner, we can sort of change the colors if we want to in the image. So for example, A T2 weighted image or a FLAIR image will give basically different colors to the tissue. So in the T2 weighted image, for example, you will see that the CSF, the sort of fluid here in the middle is white. The white matter is actually now gray, and the gray matter is still a little bit gray. So using appropriate sequences, we can sort of emphasize different parts of the brain so we can sort of get different insights and spatial information about what is happening in the brain. And that's just one part of data that we're acquiring, right? So you might be sitting in a scanner, maybe for example, for an hour. And what we do is we collect a sequence of different data modalities. So for example, another sequence that we obtain is a sequence that allows us to understand how the different brain regions are connected, the axons, the white matter integrity of the brain. So what we call these diffusion images, just because we utilize the diffusion of the water in the brain to extract these images. And we can get images like this, wherein these sort of tracts that here have different colors based on their orientation allow us to see the fidelity of the actual fibers between the different parts of the brain. And again, that's just one. Another modality that we're obtaining is blood flow images in the brain. And here it's a more complicated sequence, but the outcome of this sequence is to understand sort of the vascular properties of the brain, how blood flows into the brain and how it's been absorbed by the tissue. And as a rule of thumb for us, the higher the magnetic strength, the better the acquisition techniques, and more importantly, the better the technician and the participant compliance, the higher the quality of the data for us to obtain and subsequently analyze. And that's just for MRI. For positron emission tomography imaging, there is a different physics principle wherein you have an injected tracer that basically decays, it releases positrons. These positrons find wandering electrons, they produce an energy, and we're able to basically collect signal because of that energy. And again, similar to MRI, higher concentration of that tracer in each part of the brain will give you a higher signal in the brain. And if you use different tracers that bind in different places in the brain, we can measure different things. For example, we can use tracers such as for beta-BIR that can measure the beta amyloid, and we know that high deposits of this sort of beta amyloid peptide in the brain have been linked to Alzheimer's disease. Similarly, we can use different tracers, again using the same technique, such as for TASOPIR, to understand, for example, how the sort of tau microtubules are in the brain, again, because we know that tau proteins that sort of form into these abnormal filaments have been associated as well with Alzheimer's disease. And I will show you some of these pictures later on. Okay. So why do we use neuroimaging here specifically for Alzheimer's research? Well, historically, Alzheimer's disease was sort of basically diagnosed according to a clinical framework, right? So the presence of AD, of probable AD, was based on sort of a battery of cognitive and behavioral symptoms. But it was soon recognized that sort of, Alzheimer's disease needs to be rooted basically in biological factors, right? And the idea is that instead of sort of thinking about Alzheimer's as sort of a unidimensional problem, this approach has supported the sort of transformation of Alzheimer's to sort of a clinical and biological contrast alongside a continuum, right? That includes preclinical stages where, you have associated pathophysiological alterations in the brain before you can even diagnose, before you can even make an appropriate diagnosis of Alzheimer's at some point, right? So there's this sort of recognition of sort of biomarker signatures in Alzheimer's that neuroimaging plays a pivotal role, and also for us, it's understanding these biomarkers is essential to also optimize success for potentially disease-modifying effects, right? So if you intervene somewhere before, given these biomarkers, you might be able to understand if you have results with respect to disease-modifying effects. And brain imaging has sort of helped us immensely defining this biological framework. I'm not going to go into all of the details of this rather complicated diagram, but the idea is that there is this sort of framework that we in Alzheimer's disease we call the ATN framework, that is a sort of agnostic, a biomarker-driven classification scheme that basically relates progression of Alzheimer's disease to biomarkers as those come from PET and MRI studies. And the idea is that you have this sort of progression of the accumulation of, for example, amyloid beta peptide as measured by PET, or the accumulation of tau as also measured by PET, or other biomarkers such as CSF, as well as the neurodegeneration as measured by MRI. And sort of, I want to emphasize that this is sort of a framework, right? It doesn't imply, it doesn't imply any sort of causal mechanism, or it doesn't imply any temporal order, whether, for example, PET or tau is coming first. These are sort of active areas of research, but this is really, really important for us, because if we focus on this framework as sort of a longitudinal continuum of Alzheimer's disease, and we obtain these biomarkers at these different time points, we can understand exactly which patient, the position of each patient along this, or participant along this continuum. So this is really, really important for us. In addition, as I said, this framework is by no means complete. We and others are also looking at additional factors that can contribute to this framework, such as, for example, vascular factors. And I want to show you a little bit, I want to talk a little bit more about that, and sort of how neuroimaging provides sort of this nice picture within this framework. Here, for example, what I'm showing you on the top left, we have three sets of images. This is amyloid and tau PET, as well as the T1 weighted images. These are images that I showed you before. And here, this is a participant that has showed no cognitive impairment. And sort of this participant has been classified as, not having amyloid negative, tau negative, and sort of no neurodegeneration. And here, again, on the other sort of end of the spectrum, what we have is a participant with mild cognitive impairment. Again, the same two pictures, the same color scale, amyloid, tau PET, and T1 image. And you can see, even by sort of, I don't think I even need to put numbers here, but you can see the sort of visually the difference in these biomarkers between these two participants, wherein there is increased amyloid and tau binding in the participant here on the right, as well as some neurodegeneration in specific parts of the brain. And I'm going to come into these later on. Just again, to sort of drive this message here, when we take these images, for example, amyloid, tau, and PET, and we sort of look at different participants at different sort of levels of cognitive impairment, we're actually able to see different types of different patterns in their brain images. And you can see again visually that the sort of there is sort of a continuum wherein you have increased binding in amyloid and tau PET in participants that show some specific cognitive impairment. Another focus of our research is, one question is, does this happen everywhere in the brain? Does it happen primarily, or does it start somewhere? This sort of neuropathology, does it somewhere specifically? Well, one primary focus of us is a particular area, and this is the hippocampal area. It actually looks like a hippocampus. This is an area that it's rooted deep in sort of, I guess, the bottom and sort of deep in the medial part of the brain. This is an area that we're able to identify using those images that I've shown you before, the T1 weighted images and the T2 weighted images. And this particular area for us is really, really interesting because it's sort of a very important area for the brain for memory and learning, right? And we've seen that area being really vulnerable to these pathologies that I just described before. And getting this high quality data from the scanner allow us not only to identify the hippocampus and measure its properties, but also even go into higher, in a higher level of demarcation and sort of try and identify different subfields of the hippocampus to try and understand how different parts of the hippocampus, as outlined here by different colors, contribute, have some kind of form of neuropathology. And I talked a little bit about this. I talked a little bit about the neurodegeneration in my previous slides. And I want to emphasize that one thing that is really, really important here is this sort of cortical atrophy, specifically in that hippocampal area. And I'm not sure if you can see this movie playing in your computer, but what you can see here is that in these two slices of the same brain, this is the hippocampal area right here and here on the left. And what you can see is that sort of the worse the progression of Alzheimer's disease, basically the higher the cortical atrophy and the neurodegeneration. Basically, we sort of have, speaking really simple here, sort of a sort of degeneration, a shrinking of that particular area. And because that area is responsible for a lot of properties in the brain, such as memory and learning, we believe that that's been translated into specific cognitive impairments. One final slide that I want to show is that, for our research in this particular cohort, we are sort of looking at additional contribution. I mentioned that before. This is a study from Dr. Slakowicz's lab, again, focusing on this, sort of suspect area of the hippocampus. And what they found, it's a really interesting finding here, but what they found was that in people that carry this APOE4 genotype, which is a specific gene that has been shown to be that is basically poses high risk for developing Alzheimer's disease later on in life, they used all of these different neuroimaging markers that I described: amyloid PET, tau PET, blood flow, and anatomical images. And what they actually found was that one primary effect in these APOE4 carriers compared to controls, which in this particular case are APOE3 carriers, they found that they had higher vascular impairment. So it's not just one facet, just tau, amyloid, or neurodegeneration, but there is also this additional factor of high vascular impairment, as shown here by these higher colors in the APOE4. And this sort of was related to impaired cognition in these APOE4 carriers. So I really want to emphasize that we're looking at the brain from multiple sides, right? We're taking all of these different data sets and we're combining all of these to try and understand what is happening in the brain. And I think I'm a little bit over time. So, I just want to sort of have a closing slide here and say that using our techniques in our facilities, we can obtain high quality brain images of our participants. I really, really want to emphasize that all of the slides, all of the data that I've shown, everything that I presented so far wouldn't have been possible without our participants. And importantly, the cooperation with the technologists and all of us involved in this, I have to say, rather complicated process has been crucial to obtain the right kind of data. And if you have the right kind of data, then you can do the right kind of analysis, and you can produce the right kind of conclusions. So I would like to thank you, every one of you, very, very much for your participation. And I'm happy to take any questions regarding neuroimaging and what we do here at USC ADRC. Thank you very much.